Hey, I think we're set. Hello, everyone. Welcome to the first GRiP-ing of this season. For those who are new, GRiP-ing seminars or weekly seminars, usually on Fridays at 2, where we invite and welcome parasitologists around the world to share their cool science. We also have RAM seminars, which are internal, which is just for us, and which would really help if you could volunteer to give your talks. You could share anything. It doesn't need to be a full,
The same
full
tip that
story.
we give at Aberystwyth too. You can get volunteers.
Yes, anyway. So welcome again. That's a great turnout for the first one itself. So that's good. I'll pass on to Matt, who will introduce our speaker today.
Okay, so my very great pleasure to welcome Karl to talk to us today. I've known Karl for many years, and he's a very big name in the schisto field, very well known for his extremely collaborative lab, actually underpinning a lot of other labs in the UK, providing resources and ideas and encouragement. Karl has spent his whole career, as far as I know, working on schisto. First of all, starting off in Johns Hopkins, where he had kind of a vaccinology slant, really, working on various tegument components. Then he moved to NIAID, working on immunopathology. And then from there, moved to the Dunn lab at the University of Cambridge, and was an early pioneer of using microarrays to look at the transcriptome of the parasite across the life cycle. At some point, I can't remember when, you then went to Aberystwyth, and after you'd been at Aberystwyth for a while, became the director of the Barrett Centre for Helminth Control. That has nothing to do with Mike.
Unless John and Mike are related, I don't know.
Yeah. Well, there you go. That's slightly related. Yeah. With that, over to you, Karl. Thank you.
Yeah, thanks, Matt. Yeah, it's a tremendous pleasure to be here. I've been here a few times and overwhelmed every time I come to listen to the work that's being done here. It's a true testament to the parasitologists who are running the projects here. And more than that, it's a true testament to the people at the coalface, the graduate students and the postdocs who have had pleasure to meet today and will meet after this. And you guys should be incredibly proud. This is a premier institute for doing parasitology in the UK, and you're well known for it. And it's just a pleasure to come and listen to everyone. So thank you very much for the invitation. And what I'm going to talk about today is some work in progress. I'd love to get your feedback and get some critical comments on this because we're trying to write it up now. I think it's quite exciting because it introduces a new model, a new mouse model for looking at pharmacokinetics that is based on the humanization of the liver in this mouse. And this is what I'm considering an enabling technology. It's an enabling technology that refines our ability to do drug discovery at Aberystwyth University. Wouldn't be able to do this, so I should say this, wouldn't be able to do this without the funding, which is welcome, and without our tremendous collaborators at Dundee, University of Dundee Drug Discovery Unit, and I'll mention them in name at the end of the slide. And Andrew McDonald and his group when they were at Manchester, continuing to work now that he's moved to Edinburgh. So without those collaborators, none of the work that I'm going to present here would have been possible. First challenge. Let's just try that. That'll work. So in order to understand what we're trying to do with this new mouse model, I want to set the scene in terms of schistosomiasis. And many of you guys will know these facts, but I think this is a really good pictorial representation of what I think are the take-home messages for schistosomiasis. Highly prevalent parasitic disease, okay? We're going to know more about why it's so prevalent, I think, from the project that's being developed here by Poppy and Matt, a big welcome project that's been funded, and we're going to know why this is such a persistent parasitic infection. That number, 140 million, is a World Health Organization number, and all these numbers are World Health Organization numbers. And I think these are probably underestimates of a lot of the metrics that I'm putting here. But this is because if you wanted to look it up and to test me if these facts are right, that comes from World Health Organization. Most of those cases are in low and middle income countries in sub-Saharan Africa, approximately 95%. But we know with climate change, the intermediate snow host is being able to thrive in more northern climes. And so we know 2014, there was an outbreak of schistosomiasis in Corsica, which is in the Mediterranean. And look forward into the future, we might find that schistosomiasis is moving more northward. So be careful where you go on holiday, because it could be that you're coming in contact with snails that are shedding the parasites that could cause this disease. Again, in the World Health Organization, they indicate there's about 12,000 people or deaths attributable to this disease every year, which makes it the second deadliest human parasitic infection on the planet behind malaria. Now, I think that's a tremendous underrepresentation of the number of deaths attributable to this disease, because it's very difficult to know whether you've died of schistosomiasis unless you obviously do some pathology and other things that aren't typically done. So that's a tremendous underestimation, I think, in terms of the number of deaths attributable to this parasitic infection. And we know that there's no vaccine. Okay, so if we wanted to prevent these horrible things happening to a person who becomes infected, we don't have the ability to do so because we have no vaccine that prevents this from happening. But of course, there are people working on that as we speak now. There are 2 clinical trials ongoing, one in Uganda for TSP2, tetraspanin 2 protein, that's being done to see whether or not the very, very successful preclinical data leads to the development of a clinical pathway for this particular vaccine. But more importantly than that, we have another vaccine candidate called SMP80 by Asel Siddiqui at Texas Tech. And that clinical trial is being run in Madagascar right now. So we have 2 clinical trials trying to explore whether or not the preclinical successes that we've had with these 2 candidates can be translatable to a pathway for a human vaccine. But we don't have one now. And so all interventions in terms of trying to eliminate the subsequent pathology upon infection are facilitated by one single drug, and that's praziquantel. And praziquantel, as many of you guys will know, and I will explain a little bit in the next slide, has some known limitations. So it's not the panacea for schistosomiasis control into the future. And therefore, what we argue is that new interventions are urgently needed, and that's what drives my laboratory. So praziquantel, as I mentioned, is the actual drug that we use to give people who have been exposed and demonstrated to be infected or presumed to be infected with schistosome parasites. It's effective against all major species of schistosome that infects humans, so schistosome mansonite, hematobium, and japonicum. There's a few other ones, but those are the 3 major ones. That's what praziquantel looks like, and there's a particular carbon here, which is chiral, and so you have 2 different forms of praziquantel. You have the R form, which I'm illustrating there, which is the predominantly active form of praziquantel when you're giving a pill that has the racemic R and S. It's too difficult chemically to separate those 2 out during production. So the pill that you're given, if you're suspected of schistosomiasis, is containing both R and S. Now, the S version of praziquantel is a very bitter tasting, and I've taken praziquantel, and it is bitter tasting pill. It's part of the component of the formulation that's very bitter, which causes children to spit it out. They don't really like it. Even if you give them some orange soda to drink with it, they will spit it out. Some of them will. It depends on who's giving them the medication. But of course, knowing that, Merck has come up with a pediatric formulation of praziquantel, which is liquid, and because it's liquid, they can add in a lot of flavor, which makes it a little bit more palatable. So it is our praziquantel that is the most active, and it's only in 2021. So this drug was developed in the end of the 1970s, rolled out in mass drug administration in the 80s, and is being used in mass drug administration campaigns till now. But it was only in 2021 with 2 seminal papers, one using a genetic approach, and that was Tim Anderson's lab, and one using a more biochemical approach, and that was John Merchant's lab. They came to the same conclusion, and that conclusion was this is a target, may not be the only target, but this is a target of praziquantel, which is called schistosome mansonite protein, SMP, and some numbers. And that particular gene identifier encodes a transient receptor potential cation channel. And so all the phenotypic data that everyone has actually studied in terms of praziquantel's effect on schistosomes, the curling up of the parasite, the shaking of the parasite, the influx of calcium is due to the fact that praziquantel is activating this particular channel. Now, one of the limitations of praziquantel, and it's been known for as long as praziquantel has been developed, is very weakly or inactive against juvenile stage parasites. So imagine if you're going out into a community and you're treating people in a community with praziquantel, if they have just acquired the infection, praziquantel is likely not going to be active. Okay, so you'd have to go back into that community and give them prosopontal again, once those juvenile stage parasites develop into the adult. Now, we don't really know why juvenile stage parasites are inactive, or prosopontal is inactive against the juvenile stage parasites, but I think there's some things I was talking about today in terms of some of the staff here and Jim Collins's group, and the pharmacokinetics of prosopontal all lead to the fact that we're trying to understand this, and I think it's going to be becoming clear why prosopontal is not very active against the juvenile. So watch this space. A study that Matt's involved in, I think it's in bioRxiv now, and I don't know if it's been peer reviewed yet, or if I don't know if you got
Accepted today.
congratulations. So this is a fantastic study, a really, really, really good study, where looking at Lake Victoria and looking at the countries surrounding Lake Victoria, Uganda, Kenya, and Tanzania. What they found in this paper, and I don't want, Matt can tell us all about it, but effectively where they see treatment failures in mass drug, and so under people who are have been infected with schistosome, they're under constant barrage by becoming infected with schistosome. Treatment failures correlated with standing variation in the prosopontal target. So there's some populations of the parasite, I think this is, I'm summarizing it right, where you have the standing variation, genetic variation that leads to potentially an association with treatment failures in these populations. So all this leads us to believe that we need new compounds, potentially, unless we can come up with a vaccine, that can circumvent or help in the delivery of sustainable schistosomiasis treatment in the future. And this is what we're trying to do in Aberystwyth. It is drug discovery. In order for us to do drug discovery, we had to set up the life cycle, and this is not a trivial thing. So the life cycle that we developed is around schistosome mansoni, one of the 3 major species that affect humans, as I mentioned. And you can see here, these are pictures from either my lab or Jim Collins's lab, and this life cycle, I love it. It comes from Jim's lab, but I think it nicely shows SEM images of each of the life cycle stages. And it's broken up into three categories. You have stages of the parasite that develop in the mammalian host on the top there, schistosome up to the adult. You have free living stages that swim around the water to infect either the mammalian host or the intermediate host. And then you have stages within the snail, the intermediate host, that undergo asexual reproduction to generate loads of cercaria to infect the host. So we have access to all this in Aberystwyth, and so we can go into our life cycle and we can pull out all these intermammalian life cycle stages and do drug screens and ex vivo drug screens at any of those stages that we want. But we also run this life cycle through a mammalian host, which is the mouse. We don't infect our students. So we use mice and different strains of mice, outbred or inbred strains of mice. And importantly, by doing so, you can also study the immunopathology of schistosomiasis. So we're not only generating parasite material for ex vivo screens, we can also study what's happening when we add a compound, oral gavage compound, in mice that are infected to see if it kills the parasites or the effect. If it doesn't kill, how does the pathology develop? And the pathology develops in a normal mammal system, a mammalian system, whether it's a human or mouse, based on the fact that the females lay these eggs. These eggs get trapped in tissues, so in tissue sinusoids, such as the liver, what I have there upon necropsy of a mouse, you can see hopefully a lot of little white spots in that liver. And a lot of those white spots, all those white spots, not a lot of them, all those white spots are due to that egg becoming deposited, inducing an inflammatory response upon sectioning of some of the liver and staining with H&E or picoserous red that leads to the recruitment. So it's a type 2 driven response that leads to the recruitment of cells that induce inflammation around an egg. Because of the type 2 driven response, it induces a degree of fibrosis, and that's caused by cells producing collagen. And you can see the red, the picoserous red there is the collagen that's staining around that egg. So this is a process that leads to pathology in the liver, but also pathology, hyperplasia, that leads to the spleen that you can see there that's really, really enlarged. Collectively, this enlargement of the liver and enlargement of the spleen is what we call hepatosplenomegaly, and that's seen in the mouse, and it's also obviously seen in the human. So it's a pretty good model for studying the immunopathology of schistosome infection. And many people around the world use this as a model. So we have access to all this at Aberystwyth University, and even by having access to all these really cool tools, a life cycle, and I'm not going to show you all the ex vivo methodologies that we've developed to quantify parasite viability or motility or growth. These are all based on microscopy and robots. That's not the point of the talk I'm trying to get across to you today. We have all this at Aberystwyth. We screen a huge amount of compounds from a variety of different suppliers. So whether they're commercial, they're companies that are interested in giving us compounds, or they're academic collaborators interested in giving us compounds. We've been doing this for over 10 years. This is probably up to 700,000 screens or something like this ex vivo. So individual wells with individual parasites with different compounds. We get some really, really potent molecules, okay? And you can see there compound 26 and compound 35. These are EC50, so the concentration of compound that leads to a defect in whatever phenotype that we're measuring by 50%, okay? 20 nanomole and 44 nanomole. Praziquantel on an adult worm gives you an EC50 of that. So we're right on the same scale as praziquantel. We know compound 26 and compound 35 are equally active against S. mansoni, active against S. japonicum, and active against S. hematobium, and that was published a couple of years ago in European Journal of Medicinal Chemistry. So they're very, very potent compounds ex vivo. Subsequently, because there were people interested in microbes in my department, we screened them against a variety of different bacteria, and these compounds are equally active against gram-positive MRSA and clinical strains derived from Public Health Wales. So that was published a year ago. And then with Marcus Lee, Matt introduced Marcus Lee to me at the Sanger Institute. We sent these compounds to him, and he screened them against Plasmodium at different life cycle stages of the Plasmodium life cycle. These are active, and they're active against multi-drug resistant Plasmodium and field isolates of Plasmodium. So broadly speaking, these are quinoxamine containing compounds. It's not really the perk of this talk unless you want to look up those papers and ask me questions about it, but they're anti-infective in nature, not just against schistosomes, but against bacteria and protozoans. But the point I'm going to make is that all this really cool ex vivo data isn't translating to in vivo. Okay, so there's a variety of reasons you guys can think, I'm sure, more cleverly than me, why would you expect all this to be translatable from ex vivo to in vivo? The evidence of showing it's not translating to in vivo situations is here in this slide, where we infect our mouse model or preclinical model with parasites, and then we treat the mice at different days post-infection with 100 mg per kilogram of either compound 26, compound 35, or praziquantel, okay? Now, to get 100% or near 100% clearing of schistosomes in an infected mouse with praziquantel, you need a single dose of 400 mg per kilogram. So that's why we still see some worm burden in the praziquantel treated animals, because we're only giving them 200 mg per kilogram. But there's a significant reduction, hopefully you can see that, in the praziquantel treated animals. However, compound 35 has done nothing, and compound 26 has done something, okay? And that kind of mirrors what we're seeing ex vivo. Compound 35 is not as active as compound 26. So there's a little bit of a translation there, but it's not removing the worms as we would like to see compared to praziquantel. So we know for this particular class of compounds, one of the reasons is they're very insoluble. And so we had a whole welcome project on trying to develop the solubility of these compounds. We got them a little bit more soluble, but by getting them a little bit more soluble, they became a little bit more cytotoxic and a little bit more toxic to the animal. So we had to stop progression on these compounds. And that's a really important message, to stop something, right? Because you don't want to continue banging your head against a wall with something that's not going to work. And we've explored every medicinal chemistry. Well, I'm not a medicinal chemist, but my Dundee colleagues have assured me that they've explored every medicinal chemistry modification to these compounds, and they weren't going to get them more soluble and less cytotoxic. So we had to stop this. But equally possible, this could be a mouse-specific thing. Could it be something in the mouse that's doing something that prevents the really efficient translation of the ex vivo data to the in vivo data? And why would I suggest that? Well, a mouse, in terms of pharmacology, and I got my PhD was in the Department of Pharmacology. I know a little bit about it, but I spent many years, so I'm being informed every day by my pharmacology colleagues that what I learned back as a graduate student is not the same as what is perceived to be true today. But what is true is that metabolism in our liver, cytochrome P450s in terms of phase 1 metabolism are critical for making your compounds that you take, the medicines you take, more soluble. Then move on to phase 2 metabolism, which ultimately leads to increasing the solubility, and then phase 3 metabolism to get them out of the cell and into the urine and out into the environment. But a mouse is very different than a human in terms of a cytochrome P450 complement. It has approximately 2 times more cytochrome P450s than a human does. It has about 107 particular genes for cytochrome P450s. Humans only have about 50. So this could be a mouse-specific event that we are seeing here and trying to translate promising ex vivo to in vivo. So how are we going to explore that? We're going to explore it by using a humanized mouse model that has changed its complement of sorry, oh, that was me. So it's effectively removed all the major subfamilies of cytochrome P450s in a mouse and replaced them with the major subfamilies of the cytochrome P450s in the human. I'll take you through that. This is our enabling technology that I'm going to talk about today. It will hopefully facilitate the step change refinement. Now, a refinement is critical to the 3 R's, right? So if you guys are working with animals, this is something that we also have to consider. So this is well in line with the Home Office regulations on the type of work that we do in animals. So it is going to be a refinement according to that, but it's also going to be an advancement or perhaps acceleration in this pipeline of schistosome drug discovery, because now we can study things in a humanized setting as opposed to doing something in a mouse and then moving on to a humanized setting. So we're killing many birds with one stone here. The drug discovery pipeline, whether it's cancer or any anti-infective, is a long process. Okay, now if you were in a pharmaceutical company, you're probably billions of dollars in investment to get one candidate out of hundreds of thousands of compounds that you're screening, et cetera, et cetera, et cetera. Now, translate that to an academic setting, that could take 10 years in an industrial setting. It's probably going to take 20 years in an academic setting with the investments that we can get. So this pipeline has to be accelerated if we can. Okay, and so these enabling technologies will help do that. So I mentioned the mice that we use in the laboratory, and a lot of people use in the laboratory, or either outbred, or in this case, this is an inbred black 6 mouse. I think that's one there. If it supports the full schistosome development, I said that, it enables immunopathology to be studied, but is it an accurate predictor of human PK? And in fact, it isn't. Okay, I'll tell you that up front. It isn't, because it has too many of these cytochrome P450s that chew up compounds as you orally administer them, and I'll show you data. But can we get a humanized mouse model? Okay, so in this case, we've been working with a company up in Dundee who has made this humanized mouse model. It's the most sophisticated. It's not most sophisticated because of my face on it, but it's to demonstrate that clearly it's the same mouse, the black 6 background, but it's been humanized by cytochrome P450. But what we don't know is whether it can support the full development of the parasite. We don't know if we can study immunopathology, and we do know that it's a more accurate predictor of human PK. Pharmacokinetics based on its replacement of the mouse cytochrome P450s with humans. So what exactly do these people do to get this mouse? So you can see here a lot of X's on the graph, and these are the chromosome or the genes that encode for these 4 major clinically relevant enzyme subfamilies in the cytochrome P450s. So you have 1A on the top, you have 2C on the second line, you have 2D on the third line, and 3A on the fourth line. I'll get to the 5th line in a minute. And so Phaser Biosciences, in the seminal paper in 2019, they effectively bred a lineup of mice that basically replaced all of those ones that are crossed off, those cytochrome P450s, replaced them with the equivalent orthologous human cytochrome P450s, or replaced one for many, as you can see in the second line. And they've also replaced one for one these chemosensors, transcription factors, PXR and CAR. They replaced the mouse ones with a human equivalent. So now what we've done is we've taken 35 mouse genes and replaced them with 8 human genes. Okay, and these are the most clinically relevant genes involved in phase 1 metabolism. So that's the most important point. There might be other ones that we might be missing in terms of one drug might be processed by another subfamily. But from 2019 and not till now, they've tested these mice against a variety of different anti-cancer drugs, TB drugs, drugs that be used for human TB, drugs for different protozoan infections, and they showed that this mouse can process those drugs similar to a human, much more than a mouse can. So it's a really good starting point for us to explore whether we can use this mouse model for PK studies and efficacy studies for schistosomes. And clearly the thing is, can we bypass mouse-specific issues, reducing the attrition of what we believe are very promising ex vivo anti-schistosomal candidates to the in vivo situation? So the first thing we did was we just wanted to test whether or not the mice would be capable of being infected by the schistosome, and what were some of the responses that we saw in the host to infection. So the design we set up, we have it listed here, and you have a little box in the middle. This doesn't work. Maybe it does work. I don't know. You can't see that green. Sorry. I have a pointer. So in the middle are the 8U. So these mice are called 8U. So they're in gray, and the wild type are the wild type black 6 mice in black. And so we have in the middle of that slide, we have the animals that were infected with schistosomes, and we started off pretty conservatively. So we started off with a small dose of parasites, 80 parasites per individual, compared to uninfected 8U on the top and uninfected wild type at the bottom. And at day 46 post-infection, we took a venipuncture, we took some blood out so we can measure antibodies and other things. And then a day after that, we perfused the animals and collected all the worms, and we collected all the eggs that were found in tissues, collected the spleen, collected all the tissues, and measured recall responses with schistosome antigens, I'll show you, and a variety of other things. I can't show you everything. We've done microbiome analyses on the fecal matter of this too. That's interesting, but I'm not going to show it. If you're interested in that, I can talk about that. But the general thing here is we wanted to see whether this would support the development of the parasite. And in fact, it does. So this is a graph of all the parasitology. We're going to highlight them in sections. So the first thing we did is look at worm burdens. And yes, there are differences, but they're not significant differences. What we see is that, and generally wild type and 8U mice have similar numbers of worms. They have similar ratios, and this is important, of male to female worms. So we know from the literature that you basically have a 1.5 to 1 ratio of males to females. So you have more males than females. And we see that in the 8U mice. If you look at egg burdens within tissues, so in the liver or the intestine, yeah, there are differences, but they're not significant. There are more eggs in the liver, as you would expect, than in the intestine, and that's what we see in both strains. And if you look at fecundity, so this is basically the number of eggs per mature female, they're almost virtually identical. So the worms are developing. They are producing eggs. The eggs are being trapped in the tissues that we would suspect. The ratio of the worms, male to female bias is observed and is maintained. And the fecundity of every single female worm looks identical, regardless if you get them out of the 8U or the wild type. So we believe worm development is occurring quite normally. To further expand upon that, we did a transcriptional, an RNA-seq analysis of males and females. And we're looking at males coming from the 8U mice and males coming from wild type mice, and looking to see how many genes are differentially expressed. And that's on the left-hand side there of the vertical dotted line. And as you can see, the vast majority of genes that are expressed in the adult, which is about 8,569, the vast majority of them are expressed equally between males coming from either the 8U or the wild type. There are 3 genes that seem to be differentially expressed in either the 8U or the wild type. And that might be interesting, but the vast majority of the transcriptome profile from these worms are identical. And the same thing holds true with the female. So there's only 1 differential expressed gene at that level of significance. Now so that's p-value less 0.001 and a full change of 4, but we've even brought it down to 0.05, and we've only increased 1 or 2 more genes. So they really are similar. There's not really much going on differently in the worms. So that gave us confidence that this mouse is allowing parasites to develop normally. We looked at the immunology, and the first thing we looked at that 46-day bleed, we looked at this total IgG. And what you can see there is no significant difference between the infected wild type or the 8U and mouse that has been infected. So the 8U are the dashed bars, and the gray bars are the wild type. Now, this is a total IgG against 2 antigen sources. One is soluble egg antigen, SEA, on the left, and the other one is soluble worm antigen on the right. And again, there is no difference there in terms of the total IgG. We looked at subclasses 2A and 2B, and there's no difference between 2A, 2B, or G1. There's no difference, so I'm not showing it to you. So they are generating an antibody response similarly to the wild type. We look at splenocyte recall response. It's a little bit more complicated, but I'll take you through it. So you have SEA again and SWAP are the antigens that we are using to stimulate the spleens or the splenocyte cultures taken from these animals at day 47. And remember I said this is a type 2 driven type of infection. Okay, so what you'd expect to see when you stimulate these splenocytes from infected animals is much more IL-4, IL-5, IL-13, and even IL-10 compared to interferon gamma. And hopefully what you can see there, when these splenocytes are stimulated with SEA or SWAP, they are generating large amounts of IL-4, and there's no difference significantly between the splenocyte cultures from the 8U or the wild type. Compare that to interferon gamma, and there's no interferon gamma being produced. So this, from either of the animals or spleens from the animals. The only time you see gamma is when you use Con-A to stimulate the uninfected animals, and that's kind of what you'd expect because the infection drives the type 2 response. So immunologically, these animals are behaving the same way, or similarly. And as I said, we've done IL-17, we've done interferon gamma, we've done IL-10, we've done IL-13/5, and they look exactly like those ones that I've shown you there. Okay, what about pathology? Okay, so pathology occurs in the liver, right? And this is where the genetic modifications are occurring. We're replacing these genes. So what does the pathology look like? So we looked at it both macro and micro. Macro, you can't see much difference, hopefully, here. On the left-hand side, you have the uninfected wild type on the top and the 8U on the bottom. And then we have two examples of infected wild type in the red bar. And then underneath that, we have the 8U. And hopefully you can see there's a lot of eggs, a lot of granulomas in that. Okay, so if you look at the histopathology, looking at sections, again, the same type of nomenclature. You have uninfected on the left and infected on the right, wild type on top and 8U on the bottom. And we stain with two different colors, just like I showed you before, H and E, to show you the granuloma around an egg, and picrocereus red, which stains the collagen fibers around the egg in that granuloma. And they develop granulomas, they develop fibrotic granulomas, but you kind of have a look there at the 8U, it looks like it's a little less picrocereus red than you see in the wild type, okay? And I can see Rick going, yeah, that looks like that to me. So you can see that. And so we thought, okay, well, there probably is something occurring there that might be a little bit different. But the take-home message is that these animals can take the egg onslaught into the sinusoids, induce a granulomatous reaction, and that granuloma reaction is fibrotic. But we wanted to quantify that just to see if there was a difference there. We believe from these just very gross necropsy and histopathology, they're developing granulomatous pathology similarly. But we did quantify it. And what we found on the left, quantifying the granuloma areas or volumes, wild type animals, when you score using some automated microscopy, one of my postdocs developed, you're scoring 114 granulomas in the wild type versus 100 granulomas in the 8U. So quite a lot of granulomas are being measured here, and there is a significant difference. I mean, it is significant, but they are inducing a granulomatous pathology that are volumes of granulomas are overlapping, but it is a difference. That difference holds true, but it's not significant when you look at the granuloma percentage that contains picrocereus red. So the picrocereus red is the collagen that you see. So there is more collagen deposited in this wild type, but it's not significantly different in the 8U. So what you saw on the previous slide, when it looked like a little bit less pink or less red on those 8U mice, is validated here, but it's just not significant. What about the liver transcripts that might be driving the granulomatous response or the fibrotic response in these animals? So we looked at that next. And the first thing we looked at were the cytokines that could be driving fibrotic and granulomatous pathology. And this really comes from when I was a postdoc in Tom Wynn's lab. And this is what Monica Chiaramonte was working on, the effects of the pro-fibrotic cytokines, IL-13 and IL-4, in driving fibrosis in this model. And what you can see here, in the wild type animals, which are the black bars versus the 8U, so the wild type infected animals are the black bars, and the infected 8U or the hash bars, you can see, first of all, in the uninfected, there's no IL-13 or very little IL-13 or IL-4 message that we can see. This is transcriptome. But you can see a heck of a lot more IL-13 significantly in the wild type animals versus the 8U animals. And similarly, you see an increase in the amount of IL-4 than in the 8U animals, so the wild type animals versus the 8U, but it's not significant. So we have potentially some transcriptional validation of why you might see a little bit more picrocereus red staining. But these IL-4 and IL-13 are driving responses in the liver that lead to the production of collagen, and more importantly, the restructuring of the extracellular matrix. So we looked at genes that might be associated with that. And this is just a heat map looking, it's a little bit, I'll take you through it. So we have wild type uninfected, 8U uninfected, or the rows over top of wild type infected and 8U infected on the bottom. And we have NS, so this is a biological process through gene ontology terminology of extracellular matrix organization. I'm only showing this, we've done a variety of other gene enrichment analyses and things, but there's a lot of genes in this particular term that are not significantly different upon infection between the two animals. That's the NS. But we have a group of genes that are massively upregulated in the wild type animals upon infection than we have in the 8U. And if you look at those genes, hopefully you can see a lot of MMPs, so matrix metalloproteinases. You have ones that activate other ones. You have collagen 3A1. These are all, this is involved in the organization of the granuloma, possibly the increase in granuloma volume we see in the wild type compared to the 8U. And potentially even some more of the fibrosis that we're seeing, probably driven by IL-13 on cells that are expressing these transcripts. And of course, we have an upregulation in both animals of the cohort of genes on the left-hand side that's indicated by both or pink. So we believe that the transcriptional results that we got from the liver is really supporting the histopathology that we're measuring. So the animals now are capable of inducing a granuloma pathology similarly to the wild type. There might be some differences. The wild type might be inducing a little bit greater granuloma and a little bit more fibrosis, but the 8U are capable of doing this as well. Okay, so it's a good model for studying not only the parasite development, but pathology, and that's important. So I just said, but the most important question, can these mice be used to support our pharmacokinetic and efficacy studies? Because that's what I want to get around to. But you needed to first set up the model, right? Set up the model, make sure these guys are able to take the infection and develop schistosomiasis. So we set up an experiment, it's a little bit more complicated than the first one I showed you. I'll take you through it. So we have the infected group of animals in the middle. And what we're doing here is we're administering praziquantel, okay? We're administering praziquantel based on knowledge of data that I'm not going to show you, but we've done preliminary data looking at pharmacokinetics of praziquantel in the 8U animal compared to wild type animal. And what we found from that preliminary data is that we could administer praziquantel, the 8U animals, at an equivalent of about 100 mg per kilogram or 200 mg per kilogram, somewhere in that range, that was equivalent to what would clear parasites in the wild type animal, which is 400 mg per kilogram. Okay, so much less compound can go into the 8U mice because they are processing praziquantel differently than the wild type mice. So we administered praziquantel to the infected animals, and certain cohorts of animals also received the excipient control. That doesn't have any praziquantel. That's just how we formulated praziquantel. And we have uninfected animals as well. We took blood samples at different times post-oral gavage to get pharmacokinetic curves, which I'll show you in a minute. And then we did some antibody and spinocyte recall and all those things. I'm not going to show any of that. I'm only going to show you the pharmacokinetics, and I'm going to show you the efficacy, because that's the most important thing. So the first thing is the pharmacokinetics. Okay, so what we have here, these are pharmacokinetic curves. The x-axis represents time post-oral gavage of praziquantel, and the y-axis represents the concentration of, on the left, our praziquantel. Remember, that's the active form of praziquantel. And on the right is R trans 4-hydroxypraziquantel. Why is that important? That is the metabolite that is metabolized. So the liver in a mammal will metabolize R praziquantel into 4-trans-hydroxypraziquantel. So we know that the 8U mice are able to do that. Okay, let's take you back one step and look at the left. On the left-hand side, you have the key wild type is in the sphere, the black sphere, and then you have two triangles. 8U in black is 100 mg per kilogram, and the 8U open triangle is 200 mg per kilogram. And so 400 mg per kilogram in a wild type animal, you can see that curve. And that curve drops off quite dramatically between 6 and 8 hours. And that's what people see when they do pharmacokinetic studies, so the praziquantel in the mice. But hopefully you can see in the two two groups of animals that have been dosed, the two groups of 8U animals that have been dosed either 100 or 200 mg per kilogram of praziquantel, that plateaus off of that. And that's what you see in a human. So this is capable of replicating what we see pharmacokinetically in a human situation, even though these are humanized mice, but they are mice. And you also see the same trend in the 4-trans-hydroxypraziquantel, where you see a drop-off quite dramatically between 6 and 8 weeks of the wild type animals that receive 400 mg per kilogram, but you see more of a plateau effect of the 8U animals that receive either 100 or 200 mg per kilogram praziquantel. So that is telling us that these animals are behaving similarly to a human, despite only 8 genes being replaced, 8 genes of humans being replaced with 35, taking out 35 genes of the mouse. But another thing that gives us a little bit more confidence that this is behaving more like a human situation is in vitro data. So you can take microsomes from the liver. So the microsomes contain all your P450s. You can take them from the liver. You can take them from a mouse liver, you can take them from a human liver, or you can take them from the 8 humanized liver. And what you can see here is the clearance of prosaquantal from these microsomes. And what do you see? In a mouse, it is chewing through prosaquantal like anyone, it's gangbusters, 37 mils per min per gram. So we know, because it is 105 cytochrome P450, it is chewing through prosaquantal. But look at the human, 3.2, and what does the 8 human look like? It looks like the human. So the in vitro data is telling us it's behaving like a human, and the in vitro data is also telling us that it's behaving like a human. So we believe this model now is able to bypass mouse-specific issues in terms of pharmacokinetics. But how good is it as a model for efficacy? That's the other thing we wanted to explore. And this is the last slide. So wild-type animals, when we administer 400 mg per kilogram of praziquantel, on the y-axis is worm burdens, and on the left-hand side of that x-axis is males between the excipient control, which is 0, or 400 mg per kilogram praziquantel, and on the right-hand side of that dashed vertical line is the females. Again, something to show, to draw your attention to, that we always get more males than females. So you can see there's more males than females in the excipient control. And we see what we would expect to see with 400 mg per kilogram of praziquantel, effectively between 90 and 96% reduction in worms. Okay. Now, this is the drum roll. What does it look like in the 8 human animals? And remember, we're administering a heck of a lot less praziquantel. Okay. So the same type of format. At 100 mg per kilogram of praziquantel, we're getting about an 87% reduction in males and about 98% reduction in females. But at 200 mg per praziquantel, it surpasses what we see in 400 mg in the wild type. So we're administering less compound, which is always a good thing when you're thinking about drug discovery and efficacy. You can save, obviously, you save your really valuable drugs, but you also keep still, man. So it also allows you to effectively prevent the induction of adverse events too, okay? So we believe that this model is actually very good and enabling us to treat a mouse with levels of a compound that may be closer to human dosages. And that's kind of the holy grail of why we want to use this mouse model. So in summary, we believe this is a component of other enabling technologies that we're developing, is going to massively accelerate and refine how we do drug discovery at Aberystwyth, okay? And we hopefully will take our hundreds of thousands of compounds that we're generating really cool ex vivo data into something that we believe is worthy of clinical progression. And so we've just been funded by Wellcome. It starts in January for a 5-year project with Dundee to actually use this model and all the other models to get us a praziquantel replacement drug. So I hopefully have shown you evidence that this mouse model can support the full development of the parasite. We can study immunopathology in these infected animals. And the most important point I'd like to draw your attention to is that we believe this improves our predictive power of drug efficacy studies with more human-like pharmacokinetics. And that hopefully will refine or accelerate our pace of drug discovery as part of this project that we've been funded to do starting in January. Now, the last thing I need to do is the most important thing is to acknowledge everyone who did the work. So my group's on the left there includes Ian Chalmers, who was involved in the work. And for those of you know Ian, he's the one with the beard and glasses. And he's standing next to Remy Pichon, is a postdoc in Gabriel Rinaldi's lab at Oxford. And Gabriel and Remy, when they were at Aberystwyth before they moved to Oxford, were heavily involved in this project and generated a whole bunch of other data I wasn't able to show you, but when we write the paper up, you'll see it all then. And again, I've already mentioned Andrew, but Alice was also critically involved in generating data for this project. I think clearly all the medicinal chemistry and pharmacokinetic support came from Dundee. If you didn't know that, they're a fantastic group of people to work with. And really without them, we wouldn't be able to do any of this. So I'll leave it at that. And I think we have 10 minutes for questions, and I'm more than willing to take any criticisms, please, because we're writing this up now. If you see something glaringly obvious that we need to do, let me know. better now than after we submit. But obviously, any other questions too, I'd love to take. Thank you very much.
Thanks, Tom. Questions on the floor, and also for those online, you could just raise your hand and I can unmute you, or you could just put the question on the, oh, there's already some questions about that. But yeah, anyway, we'll first field a few questions here.
That's very busy. It's a substrate specificity of each of these, each of the deleted and inserted enzymes known. And can you reconcile the longer lasting praziquantel?
Yeah, so it's reconcilable from all the data from 2019 onwards, at least in the papers that were published. They reconcile the fact that you can get greater longevity, area under the curve, concentration of the compound in the bloodstream was reconcilable by the fact that the human version was probably had the same substrate specificity, but remember, the mouse had 2 times the amount of cytochrome P450, so there could be more of them operating on the same compound. And I think that's why you have that drop off, whereas it's more stabilized in a human. And so that's what they would explain in the papers that I've read, and likely how we're going to explain it here too.
Yes, so you replaced 35 of the P450 genes.
33 and 2 transcription factors.
Right, yeah. So that clearly had the desired effect on praziquantel. But how do you know that screening for your tens of thousands of other compounds, then the right P450
Have been knocked out and replaced.
yes,
Yeah.
yes. So you've optimized it for praziquantel, but having tested only the one drug, a cynic might say, well, you got lucky, or maybe you knew for praziquantel to target that cluster of P450s, but you'll have a lot of others.
Yeah, so we didn't engineer it to specifically, I guess, differentially chew through praziquantel. This is the way the mice came, but they have published from 2019 to 2025 studies with about 35 to 40 different drugs. So in addition, completely chemical classes different than praziquantel.
Yeah, that's what I'm looking for.
And that has shown kinetics that are similar to humans compared to mice in the 8 human mice. So you can look up the papers, I can give you the papers, but they've done anti-cancer cancer drugs, TB drugs, protozoan drugs. They're all chemically distinct than praziquantel, and they all process similarly to human microsomes and a human pharmacokinetics when it's been done. And they've chosen those compounds because they have been done in human settings. And the 8 human mice look different than wild type mice, but more similar to humans. So I think you can never say that you won't find a compound that's not gonna, you're right, there could be other compounds that are differentially processed, and I guess that's what Rick was also maybe alluding to. But for all the drugs that have been looked at so far with the papers that have been published, they seem to be processing, these 8 human animals seem to be processing it more similar to human than a wild mouse. So I think you can never guarantee it, but I think that we have some confidence that hopefully other classes outside praziquantel will be processed.
In any case, it's a good step forward.
It's a good step forward. It's a good tool. Yeah.
And I was just wondering, why do you think mice and humans have evolved to have such different levels of these kinds of?
Yeah, that's a good question. So if you think about a mouse is running around outside, exposing itself to all kinds of different xenobiotic agents, right? So it's trying to chew, it eats everything. You have mice in your attic, they'll chew on insulation. So they will eat anything, right? So they probably have co-evolved to the environment in which they live to be able to process those xenobiotics so they're not toxic to the animal. Whereas we are limited in the sources of things that we take through our mouth. And therefore the processing of the xenobiotics possibly can be done with a smaller repertoire of cytochrome P450s. Now, the one thing I will say here is that we're not looking at phase 2 metabolism or phase 3 metabolism. We are looking at it now because Russ Morphew, if you guys know Russ, is a GST expert. And one of the conjugating phase 2 enzymes in phase 2 metabolism is glutathione transferase. So we are now looking at all these livers for GST activity to see if there's any difference, or is there compensatory differences in the 8 human versus wild. We're going to look at that and see. But I think that's the reason why. It's the environment in which they co-evolve and which we co-evolve.
Yeah, the transcripts that were differentially expressed in the schistosome came from humanized mice, wild type mice, so they might be interesting.
There's 3,
Do they give you any clue about how the parasite might be adapting to the host environment?
We were hoping there would be something like that, right? Because these are worms living in the mesenteric veins that are draining the liver, right? So you think maybe if there's some difference in metabolism just normally occurring, you know, in an animal upon infection, it might change some transcriptional profiles greater than what we saw. But as you alluded to, we have three on the 8 human differentially expressed and 3 in the wild type that are differentially expressed. This is corrected for false discovery too. So we did, you know, F values, and so we believe they're real, but how relevant they are, I don't know. I mean, you know, it's one of these things that, as a side project, maybe we look at them, you know, look at why they're different. But there's only 3 in a male and 1 in a female. So, and even when I said if you decrease the p values, you only get 1 or 2 more. So broadly speaking, they're similar. But you're right, there are a few that are different, and maybe that's interesting.
Has anyone compared the transcriptome of a worm from a human compared to the worm from the mouse?
No, I don't think anyone's done that. Would you know, Matt? That's a good question. I don't think.
No. So where would you get the worm from a human?
Well, yeah. You could, well, I mean, I say that, I don't know if it's online, but I mean, there are,
Not allowed to perfuse them anymore.
no, not anymore, but people historically in Brazil.
Yeah, it was done.
It was
Yeah,
done, yeah.
it was done.
Before transcriptomes.
And whether or not the worms have been adequately, so it's a good question.
Yeah,
It's a
yeah.
good question, but no one's done that because of ethical.
Sorry, I'll just maybe
What about
ask the
me?
question online. Oh,
I was wondering about the link between the P450s and the fibrosis. Is there anything we can say about the fibrosis in humans that ties in with that? I guess you can't really directly compare it again to.
I mean, I do have a slide here. We looked at all the transcription profile of all the cytochrome P450s with the explicit aim of trying to show that the animals that are knocked out in the there's no transcripts for them.
Yeah.
But we're interested in what happens to cytochrome P450s in just wild type animals. No one's really looked at that.
Yeah.
Right, so, you know, and so what we find is that I can show you the data, if I have time, I can show it to you later, but I mean, they're minimally expressed in the liver and they seem to go down upon infection. So there's a basal level of expression, which is low, and it gets even lower when the animals are infected. I don't know what that means, and I don't know what, if anything, has been done on cytochrome P450s in infected humans. I don't know.
Yeah.
I don't know. So I don't know how to, I don't know how to bring that together. Yeah.
there's a question online from Anna Kilmado.
Oh. Hi, Anna.
Hi, Carl. Thank you for an excellent talk. I was wondering whether, I'm just reading it out
Yeah.
for people. I was wondering whether the mechanism of artemisinin on juvenile schisto is known, and if so, is that sufficiently different to the mechanism of artemisinin on malaria species? So an engineered version limiting risk of resistance in malaria would be an option?
Yeah, so Nobel Prize several years ago was given to the fact that a Chinese investigator was able to identify artemisinin as a very good antiplasmodial compound. But subsequently to that, a lot of people, including Jennifer Kaiser, the Swiss Tropical Public Health Institute, and a variety of other investigators had looked at the benefits that artemisinin derived or analogs that would have in treating people who are infected or animals infected with schistosomes. The major advantage is that it's effective against the juvenile worm. So artemisinin can kill juvenile worms. Okay, so that's a major advantage over posaconazole. A disadvantage is you're never going to do that in the field, because you would then potentially give everyone who's infected with schistosome, who is also likely to become infected with malaria, a dose of artemisinin to kill schisto, which may not be the dose required to clear plasmodium, and then you could induce many different isolates of artemisinin resistant malaria. Mechanism of action, I don't know, Anna, what mechanism of action of artemisinin has on the schistosome. I don't think anyone's investigated that.
This might not matter for your actual studies, but do these humanized mice have any differences in their sort of lifespan or health or anything?
Nothing different. So they gain weight. So we've done all the basic stuff. So they gain weight with or without infection, equally to wild type. Upon examination of all the organs after we perfuse them or infected or not, they look identical to wild type animals. The livers look a little softer. It's just, I mean, I don't know how you can measure that quantitatively, maybe there's some sort of tension you can put on the liver. They feel a little bit softer than the livers of wild type animals. Nothing else. They're a little bit more susceptible to travel. So when they sent us these animals down from Dundee, a few of them died en route or as they arrived. So they might be a little more susceptible to stress. I don't, that's just observations. I don't, so I don't know whether that, and we've communicated that to them and they said, yeah, that happened to shipping them to other people as well. Nothing else that we can see, but we haven't looked at everything, so.
How was praziquantel initially discovered?
It was discovered as it was a campaign to identify a tranquilizer, I think, for horses, and they then identified, I think it was Bayer combination with collaborating with Merck. They explored all these, at that time wasn't probably thousands of compounds. Nowadays it's hundreds of thousands or a million compounds, and they explored them against different modalities or different type of conditions that they had in the lab. And one of the things they had in the lab was a schistosome infection, and they treated mice with praziquantel and they cleared it. But it was originally developed as a tranquilizer.
Are there praziquantel is also good against other trematodes? Are there eating trematodes that could be a better in vitro screen than schisto?
I would say you could use other treatments. So praziquantel is not active against all treatments, so it's not active against fasciola. And John Merchant has identified the reason why that is. There's a pocket in the transit receptor potential myostatin channel in fasciola that has a couple amino acid changes that prevent praziquantel from acting. But because we have the life cycle and we can generate 1 million parasites per week, I'd rather go with the parasite that we were trying to develop a drug against, as opposed to a model of that parasite. In another laboratory, maybe they don't have access to the life cycle. That might be something, and people have done it with Evo-Devo type questions. It's probably better to use a easier potentially, although that arguably is not the case anymore using a planarian, because you have Jim Collins, you can do all these Evo-Devo stuff on a parasitic worm that Phil Newmark pioneered on the planarian. So, I mean, there might be reasons why you could do that in the laboratory, but for us, I think we have the life cycle. And as long as we're supported to maintain it, I'm going to continue to use it because it is the model, it is the disease that we're trying, the pathogen that causes it.
I missed what the actual screen was now against. So you're going against adult schisto that you're getting out of mice, or?
Yeah, so we do a variety of different screens from the schistosomula stage. So we can take Cercaria from a snail and we can transform them into schistosomula. And we use that as a starting point because we can generate 1 million of those parasites a week. So we can do a lot of screens. And anything that's active against the schistosomula will then be transferred on to an adult worm screen.
That's control active against schistosomula.
It is active against schistosomula. It paralyzes them, it doesn't kill them. So it reduces their motility and you can quantify that, but it doesn't kill them. If you add a viable dye, they're alive.
So you would pick praziquantel out of the screen if you were doing it with schistosomula, say today.
You could pick it out in the screen. Yeah, you could definitely pick it out in the screen. So we were provided a compound collection from Merck, blinded, and they spiked in praziquantel, and one of the few hits that we got was praziquantel from our screen. So we could identify praziquantel. We sent it back, so we only got one hit. You know, that's interesting because that's praziquantel, and all the other ones weren't hits. So yeah, you can pick it up. You can pick it up, and then go on to the juvenile stage is the final stage in our ex vivo assays. Once we get a somnol and an adult one.
I was wondering if there's endogenous molecules in the mouse that that have longer lasting life because the corresponding cytochrome is no longer there.
Yeah.
Whether those endogenous molecules might, for example, be anti-parasite in some mode of action because mice are not the natural host.
Yep. Yeah. So Kenny McLeod up in Dundee has just been given a whole load of serum samples from uninfected, infected, black 6 mice, TO mice, help red mice, 8U mice, about 5 different mouse strains that we use in the laboratory. And he's doing metabolomics to look at differences in parasite-derived molecules and the molecules of the host. Now, I don't know how he's going to disentangle. I have no idea. So we're starting to look at that. it's an interesting question, right? There might be something in that that enables us to either develop a better in vitro culture system. Or maybe an anti-parasitic because of the strain specific,
Or it could affect fibrosis also.
or it could affect
Expect
pathology. Yeah, that's good, that's a good point. So that's where we're starting. So he has all those samples, he's processing now.
I think we can close there. I think there are a lot more questions, so we can catch Carl after the talk.